Hello, everybody. In today's video, I'm going to be going over some biology, the molecular central dogma. Just gonna make sure everything's working right. So this isn't going to be important for you guys because you obviously need to understand the study of life, which is biology and the central dogma, how your DNA works. You probably had your DNA tested. Maybe you've done some ancestry DNA or 23andMe, all those kind of DNA companies that can tell you're hereditary. They use DNA technology. From crime scenes, they also use your DNA technology. And other things with fertility, they're going to be checking your DNA. We're going to go over what transcription is undergoing and how we translate to make proteins. How do we get DNA and how do we make proteins? This is going to be crucial for the things that you eat, how you exercise, and how you live day-to-day life. All of this is going to be affecting your biology. So this is going to be a general introduction. This central dogma overlies this genetic information within our own biological system. We're going to emphasize how we get DNA to all of these functional proteins, how this is going to be impacting your hormones, which are just chemical messengers that go from one point of the body to another point of the body. This is also going to impact how you eat because enzymes are proteins and they need to undergo transcription and translation. If you've yourself or have any loved ones that have had some diseases or conditions, of course, how DNA is replicated, transcribed, translated is going to be impacted by this. This presentation that we're going to be going over is going to also go over how eukaryotic organisms, which are you and I as mammals, the chromatin structure of DNA, the mechanisms of how DNA is replicated, how it's synthesized, the organelles of the cell, like the nucleus, how it's being impacted, the ribosomes, and how other things in the metabolic processes are also going to be impacted by this molecular biology course or webinar. So eukaryotic, which are mammal cells, prokaryotic are like bacteria. We are more complex as eukaryotes. We have a nucleus, whereas prokaryotes don't. We have a mitochondria that help make energy, whereas prokaryotic organisms don't. Our chromatin, this collection of DNA, is crucial for how we access DNA. And you can access it in very different ways. How you access this is going to be dependent on its structure, which is going to emulate its function. If it's tightly compacted or loosely compacted, is going to influence how it's being transcribed or not transcribed during this replication process. So we categorize this like we categorize everything else in just general. The world today, euchromatin, like you and I, when it's less condensed, it's more transcriptionally active. That's how I memorize that, like you and I. Euchromatin refers to more of the less condensed. It's not so tightly bound. It's a little bit more open. Therefore, you can structurally get into there to allow for its transcription to be active in gene expression. So then the opposite must be just where it's tightly bound, which is heterochromatin. That's more highly condensed, and this is when we want to silent transcription from happening, which is very important because we want things to be active, but we also need to know how to have heterochromatin, which is where the DNA is not as easily accessible, so where it's more tightly compact. So the importance of this is because the chromatin dynamics is very important. This process of being tightly bound, heterochromatin versus loosely bound, euchromatin is very, very important with these environmental cues that could impact that, whatever. Whether it's methylated or whether it's acetylated, which is just these chemical groups that we can put on DNA, can make things more active or less active. So this structure plays a pivotal role in its dictating of the function, which is a very important concept in biology. The structure does dictate the function. So with very specifics, the euchromatin is more or less condensed. It's ready for transcription factors like RNA polymerase and enzyme to work on it. Its relaxed configuration allows for this transcription process to occur. The opposite with heterochromatin, it's more tightly compact. It has dense regions during interphase, which is the between phases of this cellular DNA replication cycle, which is where the majority of the phase is just in between the phases. It's not meiosis or mitosis, which are just going to be how body cells or how sex cells replicate. There's high levels with heterochromatin of methylation. and deacetylated histones, which are proteins that have a specific charge that are wrapped around this chromatin, which will actually suppress gene activity or gene expression. And it's because of that methylation, that group of carbon and hydrogens bound onto it in a deacetylated histone, which is just its organic chemical structure names. It's going to be relatively bringing down that gene expression. So with this euchromatin that we just talked about, this is going to be important for transcription of these specific genes here. So this is important for chromosomal integrity, which is something that we're going to continuously get into. So with the replication, you've probably heard of the semi-conservative, like one side goes the other way and then the other side goes the opposite way of a specific DNA strand. DNA replication needs to be highly accurate. That ensures this genetic information is faithfully transmitted from the parent cell to the daughter cell. Therefore, the semi-conservative nature of a parent and a daughter cell means that each DNA molecule from one original strand, and then there is a newly synthesized strand, maintaining its genetic integrity. So the synthesis phase is crucial as it precedes the cell division, ensuring that each of the daughter cell gets the identical set of chromosomes. When you have an abnormal number of chromosomes, I believe it's called aploidy. Ploidy is like the complete sets of chromosomes. A means without. You can get specific chromosomal disorders, which you can manifest in people today. So we talked about proteins that are enzymes. There are some specific enzymes that work with that with the parent strand and the daughter strand, or just the double helix of DNA. You need to unwind the double helix because you need to have access to things. So you need to unwind it. Which a helicase enzyme, which is just a protein. So you need to make sure that you transcribe and translate this protein enzyme helicase to work. There's also an enzyme called topoisomerase that makes sure that these strands can stay apart, like it's a single-stranded binding protein. And then the enzyme topoisomerase makes sure that this helical structure doesn't overhelix. So it's important that these specific enzymes do their nature, and whenever they're not working well, or they have damage to them, or they weren't transcribed or translated well, then you might get just not good work. These enzymes might not do well. They're helping to break some of those hydrogen bonds that are between those complementary pairs of those nucleic acids. You've probably heard of adenine, guanine, cytosine, thymine. You've heard of those things, purines, pyrimidines. You can find these in just a lot of meats have purines, pyrimidines. There's a lot of protein in them. There's a lot of nucleic acid in them. They're typically in the nucleus, but they go all over the body. That's what we're thinking of with DNA. It allows to separate those strands by breaking the bonds that have hydrogen in them, because those are more of a weaker bond and they're able to be broken easily. Then this enzyme prevents that kind of supercoiling of that helical nature of DNA ahead of how the replication fork starts to work. It induces a transient break in the single-stranded or double-stranded bonds in the DNA, allowing these strands to rotate and free more freely, which can relieve some of that torsion. Because the structure dictates the function, so we need these specific proteins like this one, the single-stranded binding proteins, to play a crucial role in stabilizing to keep that DNA unbound. And it can help to protect against degradation, because you don't want degradation, because then you're going to have to start that process over again, and then you're going to have to recycle all of these degradation parts. So, when you have to paint your room, sometimes you have to add a primer. So there's a primase enzyme that synthesizes these little short ribonucleic acid primers, as DNA polymerases cannot initiate this synthesis from scratch. So it means this thing is obviously just done beforehand. It provides this OH, which is a hydroxyl group in chemistry, for the addition of these DNA nucleotides. That's important because you need that primer before you do some certain things, especially with complex organisms. This DNA polymerase, which is an enzyme, binds to this strand, and it reads it in a specific direction where you have 3-OH to the 5 direction, and it synthesizes the new one in just the opposite, from the 5th end to the 3rd end. And that's just an orientation of just a chemical structure is at this end on one of like the 5th position of something, and then the 3rd end of the position on a different organism. Sorry, not a different organism, but a different part that we give it this specific number. And it also has another enzyme called exonuclease activity, allowing it to correct errors during replication through proofreading aspects. We are more complex. We need proofreaders. You need enzymes to do their work. And this primer is done beforehand. Then you have to remove these primers from these newly synthesized DNA strands in these eukaryotes, which are just like you and I. They're more complex beings. They're mammals. This task is performed by a specific enzyme, which specifically degrades this specific strand, allowing these systems to just utilize this resulting gaps in deoxyribonucleic acids. The deoxy refers to it does not have an oxygen in that DNA because it's deoxy. But you also have ribonucleic acid. And you need to make sure an enzyme, which is DNA ligase, kind of glues and seals things together. It performs these phosphodiester bond groups, phosphate di to ester groups between these adjacent nucleotides. Nucleotides, a nitrogenous base, a sugar, and a phosphate group. This effectively seals these things together to ensure that the integrity, and again, the structure, is what dictates the function in that enzyme. So now we talked about those two strands. One is a leading with a specific number, then one is a lagging strand. One is a continuous synthesis without any interruptions. That's the leading strand. Then there's a lagging strand that's just not completely continuous, which is why it has the term of a lagging strand, because of these specific fragments that are in there that I believe some Japanese gentleman found, Okazaki, then called the Okazaki fragments. Fragments are these things that kind of get in the way. Therefore, it's discontinuous. Now, to transcribe from DNA to RNA, there's different kinds of RNA. It's a process of an RNA synthesizes a messenger RNA from a DNA template in the nucleus. So transcription occurs in the nucleus, which is like the CEO of the cell. It's copying these gene sequences into RNA, which does not have oxygen, it's ribonucleic acid, before translated into proteins outside of the nucleus. So now this processing occurs from an enzyme. It binds to something that's promoting it of the gene to help unwind that helical DNA. And it's reading from the 3 to 5, and it's synthesizing from the 5 to the 3 direction. This process does not require a primer. Then you have to read this template strand. In transcription, it carefully reads an antisense or a template. I like to think with the T's for anti refers to template, and then sense is the coding strand of the DNA moving in the 3 to 5 direction. This orientation allows for the enzyme to synthesize the complementary molecule from the 5 to 3 direction, ensuring accurate transcription of the genetic code. So far, in just, I think, everyday non-scientific language, the structure of these directions are important and are highly regulated to be able to transcribe specific proteins, etc., which make up a huge part of our body. This is a structured process. There are these things of these strands, introns and extrons. Some have coding, some are specific for coding, and some are like recycle parts. So we call this a spliceosome. Is a complex of a snRNA, small, I believe SN is for small nuclear RNA, and then a protein that plays a critical role in the messenger RNA. It recognizes specific sequences, introns and extrons, facilitating the removal of introns and the litigation of exons, which can result in distinct mRNA transcripts through alternative splicing, thereby which increases protein diversity, the introns and the exons. So you could do other things that modify things of these things. So you can have what we call like a 5 cap, and I believe it's like a cap to the 5 end of things, which has a guanosine molecule. The 7-methylguanosine is added to the beginning of an mRNA to prevent it from degradation and assisting in ribosomes binding during translation, which occurs in the cytoplasm of eukaryotic cells. So you add a poly which refers to many. A, I believe, refers to the adenine or adenosine, consists of a long stretch of adenine nucleotides that enhances the stability and facilitates the export of this messenger RNA from the nucleus, which is where transcription occurs, and in the cytoplasm, which is where translation occurs. And these are in separate times in eukaryotic cells. So then you have to decode this. Translation occurs from the messenger RNA, is decoded by ribosomes to synthesize polypeptides, many peptides. This is essential for involves multiple factors like transfer RNA, which is tRNA, ribosomes, and various enzymatic activities to ensure that these proteins are produced correctly. They're produced in the cytoplasm, but we have to go from the nucleus. So you have to get outside the nucleus to go into the cytoplasm, which is the jelly-like fluid portion of the cell, where translation occurs. These ribosomes, there's ribosomal sites in the cytoplasm. And we had messenger RNA, you have a transfer RNA, and you have specific sites. An A site, where there's an amino acyl group, this is the entry point. So the A is the beginning point where the transfer has to start to transfer this. And it brings in a specific amino acid that helps to start this coding process. You have to make sure that that specific positioning is correct. And then there's a specific coding sequence that helps to start things. It's where we're talking about RNA now because we've gone DNA into messenger RNA. So now you're going to have a uracil, you're going to have an adenine, you're also going to have a guanine. Adenine, uracil, and guanine, and that specific sequence is the start coding sequence for this transfer process. So during this translation, you actually have peptide bonds that are here linking these amino acids together. When you link amino acids, that's a peptide bond to make a peptide. As these ribosomes are going to be moving along this messenger RNA, you're growing the peptide chain. That's why it's called the P site. And then you have an E site for discharging or exiting site. This is where the transfer RNA is not charged now, and they've donated their amino acids, and they're released from this specific ribosome. This is important for recycling, for future processes to occur. So now we talked about the specific coding sequences, which are typically a few of these nucleic acids. There's so many different kinds of codons. I believe 64 possible codons, which are derived from nucleotide bases that are adenine, uracil, cytosine, and guanine. So those codes to help for specific amino acids. Our body uses those nucleic acid codes like a computer uses a 1 and a 0 coding. We're just trying to code for specific things. The starting codon actually starts for the amino acid methionine. Methionine is a sulfur-based amino acid that's found in, I believe, various meats have methionine, and it's incorporated into a lot of proteins. These are the coding sequences that allow for the stopping of that specific protein. It's UAA, UAG, UGA. These actually don't code for any amino acids at all. They actually signal for this termination of translation. And then they have to recruit specific release factors just to make sure that these newly synthesized proteins, these processes need to go disassembled. So these are coding sequences of these 3 nucleotides. They determine the amino acid sequence, which is going to be important for the specific coding structure. This dictates the correct amino acid that are growing to this polypeptide chain. There's a hypothesis that there's a wobble hypothesis that the third nucleotide of a codon is not necessarily as it doesn't have to be 100% exact or specific. That in general is hopefully a little general mastery of some of the central dogma of molecular biology. I'm going to show you guys some more specific aspects of science here. So hope that definitely helps. I'm going to find some more specific scientific topics that I have pulled up. So I have many specific and different webinars that I've created with specific PowerPoints. Hope you guys are enjoying the day. Hope you guys are enjoying the scientific topics here. All right. Okay, this one's loading here. Thank you for waiting. I'm gonna add this here. There we go. Okay, so the goal with this one is obviously a lot of our body is protein. So a lot of people need to ensure that you have a good understanding of protein in general. So this is going to go over specific protein structure. That way you guys have a good understanding of how proteins are being formed, how they're being made. Protein structure is really important because it's virtually in all specific processes, like what we talked about. Whenever you're eating food, you're going to be having proteins. It's a part of the enzymes. You could take enzymes as a supplement, but enzymes are going to be crucial for just breaking down so many things that you eat, so many things that your body creates, like hormones, neurotransmitters. These things, hormones and neurotransmitters, they just transmit one area of the body to another area of the body, some kind of messenger. Like an example for you have so many hormones that come from a pituitary gland by the head region that goes down to your reproductive organs. You have things that come from your throat, from your thyroid gland that help with energy metabolism. Metabolism is defined as the sum of all of the chemical reactions in your body. This is crucial for your energy. This is crucial for your ability to actually build tissue being anabolic, but also your ability to, like, when you burn energy, where does it actually go, right? Whenever you're burning things, you're actually excreting it through what you breathe, through the urine, through some of the fecal matter, through some of the sweat. And this specific protein, they're found everywhere in the entire body. So there's a primary structure, which is a linear sequence of amino acids, is just the first primary thing. It's not complicated. It's not a three-dimensional structure yet. It's just a linear chain of amino acids. These are linked by peptide bonds. Probably heard of peptides like insulin, glucagon, or peptides that you could buy from these research companies, like these glucagon-like peptides, the GLPs in 2026, that have been very popular for many years now. And these are extending from an amino group of an amino acid to a carboxyl group of amino acid. So it's a specific sequence from the amino group to the carboxyl group, and they have a specific amount of protonation or deprotonation groups, depending on the charge of the amino acid. Proteins fold into more specific protein structures, like a secondary structure. These involve alpha helices. Helices are in that kind of curved pattern. There's also something called a beta pleated sheet. These structures are stabilized by hydrogen bonds. Like the meat you're eating or the protein you're having from a plant-based diet, you're going to be having primary structure of proteins, secondary structure of proteins. These are playing very important roles. Certain amino acids have specific structures to them. There's, I believe, 20 amino acids. One of the amino acids is it's kind of different. It's an outlier. It's called proline. It's found a lot in our collagen in our body. Collagen is a very abundant protein. Proline has a specific kind of structure to it, where part of the backbone of the amino acid, which all amino acids have a backbone, it has like a circular ring around its backbone, and that makes it very specific that it can be in certain proteins and it can't be in certain proteins in specific areas. So now the tertiary structure, tertiary for three, it's a three-dimensional structure. This structure is stabilized by various specific interactions: hydrophobic, where they don't like water; hydrogen and ionic interactions; and covalent, sulfur bridges. This is very critical for protein function, denaturation, which is just the breakdown of specific structures. Then the more complex is a quaternary structure where you just have multiple chains of proteins, multiple subunits of proteins, like bigger proteins. This is stabilized by the tertiary protein structure bonds we talked about, and they add for different diversities to bigger proteins. Like a very common example of a protein would be hemoglobin. It has iron in the center and it has just a big protein. That's for your blood. You test your hemoglobin A1C, you're testing a lot of stuff about hemoglobin. Those are the primary structures, secondary, tertiary, and quaternary. Primary more simple, quaternary more complex. So this is going to dictate its primary structure and its function and its functional properties. Insulin and glucagon are peptide hormones. Primary structure does not necessarily get degraded with specific like heating of things or specific things that can break down proteins. They typically break down the protein structure of quaternary, tertiary, and secondary, but typically not the primary structure of proteins. So these are the secondary protein structures, alpha helices and beta sheets. Alpha helices are coiled, dynamic, that are stabilized by hydrogen bonds. There's an amino acid. There's many amino acids. This is necessary for its stable structural protein functionality. A different structure is beta pleated sheets. Their parallel or even anti-parallel arrangement allows for greater stability versus just a linear nature of these hydrogen bonds. Structure is important for specific proteins because when proteins don't have specific protein structures, they can be read differently or they can actually impact the day-to-day manifestation of what these proteins do. So if you have abnormal folded proteins, especially in the nervous system, your brain, it can be very detrimental. So tertiary structures, like what we talked about before, they're going to be more complex. They require more collectively stable protein shape that would dictate its overall functionality. And then quaternary protein structures, like in the picture, it shows you the different colors. Hemoglobin is a really good example of this, but there's other quaternary protein structures that have kind of different complex things that make it up. So specifically, the direction matters of the proteins we talked about. Polarity is like a positive and negative pole. Everything has charge to it. We talked about the amino acid backbone. It's crucial for protein folding and how it dictates these amino acids are going to be interacting with other things. So we talked about enzymes. Those are proteins. In enzymes, we've talked about the specific protein structures, and there's different ways to structure the protein. Different amino acids have different charges to them. Some like to interact with water that are more polar. Some are non-polar that don't like to interact with water. So the protein is going to be dictating which amino acids are part of their protein. They orient them in a specific order that makes sense for what that protein wants to do. And if this kind of protein folding isn't done well, then it's going to be negatively interacting what the protein is dictated to do. And it's typically related to its polarity, negative charge, positive charge, or no charge at all. So where things are being held in a protein is very important. For when people come into probably a doctor's office and something's going wrong, something could be going wrong with how their proteins have been made for quite some time now. This can impact just your body's ability to build energy. For people that are wondering why they can't gain weight, something like this might be occurring in your body where the protein structure isn't being structured well, it's not functioning well, it's not being laid down well, the amino acids might not be oriented in the direction that they need to be, etc. So hydrogen bonding is done everywhere. You have a carbonyl oxygen group and an amino acid group. You need to have nitrogen, which can come from just the environment and from food. Same with carbon. You get this from your food. Nitrogen-containing proteins, obviously, are going to be meats are good protein sources for people from mammals. Plants have good protein sources, but they're typically incomplete proteins. You have to combine different plant sources to get a complete protein where you get your nitrogen from. So this bonding pattern is necessary in that helical nature. And if it's not, you're going to have some issues. So, of course, the structural protein is necessary is very important. Specific amino acids called proline, because of its specific structure, like what we talked about before, it can actually cause an issues in these specific proteins to fold due to its kind of circular ring backbone. The specific amino acids, cysteine, is also unique because it can help form these big sulfur bridge bonds that are very, very strong. You're going to find some of this in your hair. Is some of the kind of that nature of sulfur and kind of keratin as well. So, quaternary protein structure groups and its cooperativity. Let's see. Quaternary structures are involved in these multiple polypeptide chains. That's a quaternary protein structure. Instead of these encoding these separate genes, each subunit, a single gene can be used in multiple copies, thereby minimizing DNA coding requirements and the potential for errors that are occurring when we get translation of proteins. So, cooperativity of these things, whenever you bind a ligand, which is just material, like when oxygen goes on a hemoglobin with one conformation of the subunit, it can help bind the other ones as well. Like it's cooperative binding. Once one thing binds, it starts to help with the other ones to bind, like a domino effect. Thermodynamic of food, thermodynamic of protein folding, where obviously heat is going to be involved, the hydrophobic effect, which doesn't like water. It's as proteins fold, these hydrophobic ends or residues are buried in their interior because they don't like to be surrounded by water, and most of your body is water, so they have to be away from water, which can significantly increase the entropy or the random or disorder of anything, of the surrounding water molecules, which leads to a more stable protein structure because of the hydrophobic core. A Zwitter ion, I believe Zwitter is a German word for hybrid. We talked about the amino group and the carboxyl group, which can be a positively charged amino group and a negatively charged carboxyl group. It means you're going to have like a positive end and a negative end. It's a dipolar ion. It's a molecule that has two poles. And there's a point at which there's a neutral charge at around equilibrium. Iso refers to it's not moving. It's the isoelectric point. We've heard about pH before. It is the power of hydrogen, in which a protein doesn't exhibit any kind of charge. Therefore, it's a hybrid. The charges just neutralize each other. At this point, the number of positively charged proteins should be equaling the negatively charged proteins. The isoelectric point, we're going to be varied via different proteins and different pHs. Negatively charged amino acids are going to have a different one than a positively charged amino acid rich protein, etc. Some proteins have more positively charged amino acids like lysine or arginine, and some are going to be having more negatively charged amino acids like aspartic acid or glutamic acid. Their isoelectric points are going to be different, and that's of course going to be affecting their behavior within the specific proteins, within the DNA, with whatever they're interacting with. So it's getting down to very nitty-gritty specific details. In a specific hospital setting or lab, they need to detect these proteins. And I believe this is even how protein powders can be made, etc. This technique helps preserve the structural nature of proteins, allowing for enzymatic activities and protein interactions. It's called a PAGE. I believe that refers to like a polyacrylamyl gel electrophoresis. Electrophoresis is a charge. SDS page uses a sodium dodectyl sulfate to denature proteins, like break them apart, which is going to be imparting and opening a specific charge proportional to their size. So this is how you can change or find out the charge and the size of proteins, and you can separate them based off of their size, of their molecular weight. Some proteins that are smaller are going to migrate faster through this gel of things of how they're testing the proteins. Then you could focus on their isoelectric points. You could dictate them and test them based off of in a pH gradient gel. Proteins will migrate until their pH matches their isoelectric point. Then they just stop. So that's how they could test some of these proteins at what they're specifically doing, how much their size is, what their charge is. That's how they could test. So other important proteins, of course, you talked about hemoglobin. Let's see if you can see this in particular. You have hemoglobin and you have myoglobin. Globin, these are big, big proteins. Heme has heme, like heme iron. Myo is going to be, of course, in the muscle. Hemoglobin has a cooperative binding mechanism like a domino effect. When one binds, the others start to bind via a tense state or a relaxed state during oxygen binding. This is important for obviously people with, you obviously need oxygen to be healthy, but if you have too much carbon dioxide, it's going to impact your hemoglobin. Like carbon monoxide poisoning, for example, can affect hemoglobin cooperative binding, so that which can kill you. And then myoglobin displays not cooperative binding, so it doesn't have that domino effect. It does not exhibit an increase in affinity or stickiness after a first oxygen binds. Myoglobin serves as an oxygen storage protein in muscle tissue rather than this oxygen diffuser, which is different than hemoglobin. So we talked about specific protein structures. When they're structuralized well, you have healthy tissue. When you don't, and especially when these proteins clump up, or they plaque together, they get sticky. Some of this can impact your arterial health, like plaques in your arteries potentially. It's very much so going to affect your brain tissue or the brain cerebral arteries with these plaquings that might be found in these neurological conditions that affect just the function of the brain, like speech, memory issues, cognitive processing issues. These are the dementia, the Alzheimer's, potentially the Parkinson's. When you don't have these proteins structured well, they don't function well. And you can clearly see it within a human that's been functioning for a long time, but then as gradually they start to decline with so many things, their mood, their memory, their smooth movement. They start to get trembling. They start to not just be able to grow tissue. They start to become very catabolic and skinny. They don't have an appetite, etc. They don't sleep well. All of those things are what the brain does. So if you don't have healthy protein structure, it might be impacting that. Prion disease is more of a rare condition. I believe you might get some prion diseases or Creutzfeldt-Jacobs disease, misfolded proteins. Prions, you might get them from pretty bad meat, I believe. So you probably heard of like insane people that do these crazy marathons. I've had some patients, chiropractor patients, that have done ultra marathons, which is like 60 plus miles. I mean, just super intense, running 20 hours a day, right? If you get too much muscle protein breakdown, it can lead to a negative balance. It can lead to muscle loss. It can lead to kidney issues. It can lead to a ton of issues. Okay, so how do we get protein as an energy source? You consume protein. You can also get them from obviously amino acids are proteins. You can take these as supplements. You can also get the glucose can go to an amino acid called alanine. Glucose is a sugar. It's transamination. So trans across amination in muscle. During prolonged exercise or fasting, you can get skeletal muscle can undergo a transamination where you get trans refers to across. Amine is an amino group in amino acids. They're converted to this process, ketoglutarate, forming glutamate in the process. This reaction facilitates removing nitrogen from an amino acid that transports them for metabolism. Breakdown of muscle into sugar. The alanine that's produced in the muscle enters the bloodstream, transported to the liver. This is essential for having a healthy liver, but also getting nitrogen from your muscles to make actually sugar energy, which is used for energy. So yes, you can break down your muscle into sugars. And then you convert it into urea, or you can utilize it for making new sugar, which is gluconeogenesis in the liver. Alanine goes into pyruvate. We've probably heard glycolysis breaks down pyruvate by alanine aminotransferase. An enzyme transfers an amino group from alanine, which removes the amino group. Then pyruvate goes to make new sugar, all that good stuff, because you need healthy amounts of sugar. So we talked about nitrogen balance in specific plant proteins, animal proteins. You could take essential amino acids, which have a ton of different amino acids. Branched-chain amino acids are a bodybuilding supplement. Leucine, isoleucine. Methionine is really important. Tryptophan is important for sleep, mood, etc. All that stuff. So people might check their nitrogen balance. I don't even necessarily know how you could check that, but a positive nitrogen balance indicates that the protein synthesis exceeds breakdown, which is why it's positive. But if you have more breakdown than synthesis, it's a negative nitrogen balance, which is found in more severe starvation, severe illness, malnutrition, etc. Protein is very important for you. So the starvation biochemistry and prolonged exercise, prolonged fasting, you're going to have gluconeogenesis. You're going to deplete your glycogen. Fasting can be helpful. During early stages of fasting, you're going to break down some of your glycogen. You might be making some ketones by now or in the future. This relies heavily on amino acids that are released from muscle tissue, which is why people, typically like athletes, eat pretty frequently because they don't want to break down their muscle tissue. You can make ketones that are come from acetyl CoA, that come from fat, that can help make energy. Okay, so things that are also going to be occurring is you can use phosphorylation with phosphate groups to specific amino acids like threonine and tyrosine and serine. These are amino acids. Due to their structure, they have a hydroxyl group. A hydroxyl group has the ability to have a regulatory switch that can help phosphates attach on via phosphorylation, and the removal of phosphates are done by phosphatase enzymes. Serine and threonine and tyrosine are the main amino acids that are going to be phosphorylated, and that's really, really, really important. So the process of glycosylation is an enzymatic process where, of course, you have a sugar attached to a protein, like hemoglobin A1C. You have too much sugar bound to a protein. Sugar is kind of sticky. It could bind to a protein. Too much of this is not good. But glycosylation is also important for cell signaling, recognition, and immune responses and how proteins can interact with other molecules, and that's due to the protein bound to the sugar. And this is how a lot of the immune system can recognize certain things. With specific proteins, there's obviously different kinds of proteins. Obviously, muscle meat is going to be different than like a specific, protein from a bean. Their DNA is obviously the same, but how collagen is, it has different tensile strength than hair, skin, nails, keratin. Globular proteins are like more spherical in shape. They're more soluble in water. And that's going to be different than like more fibrous proteins. So obviously the protein you eat matters, but it's recommended to generally have a wide array of different proteins. Especially as we age, it can be definitely tough to synthesize a good amount of proteins. There's muscle wasting disease, sarcopenia. Not exercising because exercise is a good stimulator of protein synthesis, growth hormone, etc. And a lot of us do not want to have these neurodegenerative disorders. So you want to have healthy protein. You want to make sure you exercise. Like resistance training is going to be very, very good. For muscle synthesis, you're going to have endurance. Endurance activities is also really, really good. For aerobic capacity, protein science and medicine. Is it helpful to take protein supplements? It might be for you. Maybe amino acid supplement can be helpful, or eating healthy proteins from animals and from plants. Making sure protein folding is really, really good, which I hope you learned a lot about that today. With aging, we want to ensure that we have healthy protein levels throughout our entire life. So for me, I like to exercise a lot. I like to consume healthy amounts of protein. Nowadays, with a lot of these fitness trackers or even language learning models, you can take a picture of your food, upload it into free apps like Google Gemini, ChatGPT. You can generally ask it, how much protein do you think this food has? What are the types of amino acids found in this protein? And give it your body weight and just see what it says to see if it's something that can be helpful for you. For some people that tracks their protein, it can be helpful. Now I'm going to show you guys other specific. Awesome. So I'm going to show you guys some more specific PowerPoint presentations that I have that I think can be very, very helpful for some people. All right, I'm going to share the screen here. So I think this is a topic that honestly can be confusing for some is what pH is. It's the power of hydrogen. You've heard of alkaline water. You've heard of acidosis, too much acidity, alkalosis, too much alkalinity, etc., which is the opposite of acidity. This is obviously going to be really important. You could test pH via like a little dipstick thing, dip it into a water solution and see how much hydrogen there is. Obviously, the pH is going to be impacting a ton of your body because it's just the general concentration of hydrogen in a specific solution, like your blood, which is going to dictate, can it break down protein? Your stomach acid is very acidic, and it's going to break down specific proteins. It stands for measuring the concentration of free hydrogen ions in a liquid, which indicates how acidic or basic a specific substance is. The acids that are low pH, which because it's the pH spectrum, you probably heard it's a negative spectrum. So acids have a lower pH, bases are a high pH. Acids, they like to give protons, they like to give hydrogen ions. Often sour things are more acidic, that it's more reactive. Bases, a higher pH, which is going to be more like soap. They feel more slippery and they have they're takers of these hydrogen protons. Around neutral is around 7 because you have 0 to 14. So the halfway point is going to be 7, which is going to be closer to just basic water, which is why the companies that sell specific waters, more alkaline, more etc., is a perfect balance of hydrogen protons and then hydroxide or OH ions. You've probably seen stuff like these as little chemical structures. You have obviously a hydrogen, which are going to be the two ears of the Mickey Mouse. And then because it's one oxygen, the oxygen is larger than the hydrogens. If you imagine a periodic table, hydrogen is on one side, then the oxygen is going to be on the other side. Oxygen is larger, and that's why it's the red thing here, and the hydrogens are the white ears. Those are typically smaller. On the periodic table, hydrogen is at the very top left. So water is constantly undergoing self-ionization, which ionization it splits into the hydrogens and the OH hydroxide ions, which can increase in free hydrogen concentration, making a liquid acidic. Like your stomach acid's very acidic, your blood's not very acidic, which is more basic. This logarithmic scale goes from 0 to 14. This means each whole number change represents a 10-fold difference in acidity or alkalinity. For a pH of 4, it's 10 times more acidic than a pH of 5 because it's a 10-fold every 10th. It's like 0.1, 0.10, 0.100, etc. Proteins are obviously very essential for life. Their folding, it's going to be dictated to how sensitive they are to specific pH because the hydrogen ions carry a charge. Extreme shifts in pH can cause the unfolding of proteins, can cause them to denature, which is why obviously pH is really important, whether that area of the body is more alkaline or whether they're more acidic, etc. A big thing is our body's buffer and resistance to pH changes. These are buffers. Bicarbonate is a very common buffer, like sodium bicarbonate. It's the buffer is a chemical, like a seesaw that can absorb excessive hydrogen ions or release them as needed, which can help to resist changes in pH. By exhaling carbon dioxide, it's like we are literally removing acidity, keeping that blood pH pretty regular between 7.3 and 7.45. pH is a highly sensitive thing. I don't know how much of whether the food or the stuff we eat directly impacts the blood pH, but it's probably going to indirectly impact it because of how tightly regulated this is. Can your breathing impact the power of hydrogen in specific areas? It seems like it can, but only by a little bit. There's an enzyme called carbonate, anhydrase, the body's pH regulator. It's going to facilitate the reaction of carbon dioxide and water to bicarbonate to change just HCO3 and H plus free ions in tissues. This can help convert that carbon dioxide. You don't want to have too much carbon dioxide. It's not healthy, so this enzyme needs to work, and it's mainly primarily in the lungs. So in the lungs, this reaction reverses, allowing carbon dioxide to be exhaled, which can regulate your blood pH. So carbonic anhydrase, the lungs, blood, and carbon dioxide and pH. So, yes, do we think that eating alkaline foods can significantly change your blood pH to prevent disease? Your blood's efficient buffer systems, like the bicarbonate system, is what really regulates the pH, regardless of the diet. If food changes your blood pH, you could be critically ill. Food primarily affects the urine pH, not necessarily the blood pH. But marketing is going to obviously say otherwise. More companies that market things. To give you an example, very acidic is your stomach acid. It can break down things a lot. So the power of hydrogen ions can help to denature proteins critically, which is why it's important to have a healthy acidic stomach because you need to break down specific things. The skin, a little bit more acidic than your stomach. The skin, around 4.7 to 5.5, it's the acidic nature of it is good to break down things like bacteria that come onto our skin. The small intestine, just after the stomach, is going to be more basic than the stomach acid. It neutralizes stomach acid for nutrient absorption after it's been broken down in the stomach. Then the blood is going to be even more basic than the small intestine, and it's 7.3 to 7.5. So here's some pH. So I'm having some coffee here. I had some tea earlier. Coffee is naturally acidic. Obviously, it depends whether you did hot or cold because the heat and stuff can break down things where the cold that necessarily doesn't. Hot coffee, more acidic, which is not necessarily bad. Cold brew is going to be less acidic because the heat extracts more of these acidic compounds. Heat is a good extractor. As we obviously know, it can burn you. Teas are generally less acidic. Herbal teas are even often near neutral pH, especially again if it's cold, hot, etc. I also recommend that you don't use tea bags. You don't want to heat up the bag compound, so just have loose leaf tea and not straight from don't have Keurig coffee because that heat could probably leach some of those plastics into your drink. Same with tea bags, don't have those. Alkaline water, like mineral water you guys can buy from the store. It's naturally alkaline because it's water. Distilled purified water can become slightly more acidic by absorbing some carbon dioxide in the air, forming carbonic acid, which is not necessarily bad, like carbonated water, etc. Your specific areas of your body have different pH. The mouth pH is going to be different than the stomach, etc. Your mouth is where digestion starts to begin in the oral cavity. Bacteria in your mouth can metabolize sugar and produce lactic acid. This can drop that pH, which can start to enamel and erode on the tooth enamel. So it's going to obviously impact the levels and health of your teeth. Continuous sipping of acidic drinks keeps your mouth in this very damaging, eroding teeth stage. Which is why you probably heard you probably should use like a straw. You probably heard of ocean acidification on a global scale with too much atmospheric CO2. We know CO2 can obviously cause the area to be acidified. By absorbing a carbonic acid, it lowers the ocean pH. Ocean acidification, this ocean becomes affecting the marine life, shellfish, organisms in common places. I used to live in Hawaii. I know people from Australia are really big on this in the coral reef, etc. This is going to affect the calcium levels of specific shells and skeletons of our marine life. So these pH things, okay, this is a little tip here. Nature's pH indicator, if you boil red cabbage juice, you're going to turn that red and pink in acid, but it goes green and yellow in a base. If that makes sense. Amino acids obviously have their building blocks of proteins, like what we talked about earlier. They have an amino group and an acidic part of it. All right, that is that specific presentation. That's really important because for some people, because pH can be just can be a difficult thing for some people to understand. So now I'm going to be doing some more specific topics and help you guys with your specific science knowledge. This in general can be helpful for people that way. They have a better understanding that way. They're just not listening to people without any kind of general scientific knowledge. So I'm going to go over some specific metabolic pathways. This is how our body likes to make energy. Sorry about that. Okay, so understanding of some of these glycolysis and electron transport chain, how your body metabolizes energy. The metabolism is the sum of all of the chemical reactions in the body. These biological pathways, like the breakdown of sugar, which is glycolysis, and how we can transport electrons, which occurs in the mitochondria, glycolysis is in the cytoplasm, they operate under non equilibrium conditions, ensuring a continuous one way flow of these reactions. The body maintains what we call a negative delta G. Deltas change by constantly consuming products and supplying reactants via kind of a balancing principle. But there are steps in glycolysis that are irreversible, that are just really crucial. These are the factory regulatory points where you have managers at these specific parts of the factory that are just really important. That these managers want to make sure that these steps they go really well. You have hexokinase, one of the first steps. It catalyzes it's phosphorylating glucose. You have an enzyme hexokinase in certain body regions and glucokinase in certain body regions. They help to catalyze this first irreversible step, which is just going to be phosphorylating glucose. Steps 1 is hexokinase. Steps 3 is we have an enzyme, PFK1 or phosphofructokinase 1. We have glucose, it gets phosphorylated, it gets isomerized into fructose, which is just changing the chemical orientation of glucose to fructose. This is a rate limiting step where the managers they're there making sure the step works because you can't go backwards from here in the factory, you know what I mean? Then the steps 10, pyruvate kinase, glucose goes to pyruvate, catalyzes this irreversible step, generates pyruvate, and then 2 ATP molecules. These are major metabolic pathways. We have glycolysis. In the cytoplasm, you get ATP, you get pyruvate, you get an electron carrier, and a DH. The Krebs cycle, which occurs in this mitochondrial matrix area, the mitochondria has two membranes. Mitochondria is believed to come from bacteria in our body. It's an organelle that helps with energy. You get these electron carriers. You get NADH, which comes from vitamin B3, niacin. FADH comes from vitamin B2, riboflavin, for carrying electrons to make energy. We need oxygen for this step. In the inner mitochondrial membrane, you have oxidative phosphorylation, which generates ATP via this proton, like hydrogens, this specific gradient to make energy. We can also oxidize fats in the mitochondria, which is why fats are not bad, but you need a healthy mitochondria to break down fats. So you have coupling effects, you have metabolic pathways, release energy. Of course, whenever you're working out, you're going to, yield energy, yield heat. Whenever you're eating food, you actually have a thermodynamic effect of food because you're breaking down food, and the body cleverly couples these exergonic reactions with energonic reactions, is known as spontaneity and coupling, such as making ATP synthesis, like a waterfall and a power mill. Like a waterfall is using kinetic energy to power a mill. Regulation of enzymes are controlling the rate of thermodynamically favored reactions, which can help with breakdown. Certain things of these metabolic pathways, they create energy, but if you already have a ton of energy, then you actually are going to inhibit these metabolic pathways from occurring. There's a principle called Le Chatelier's principle, which is just a balancing effect, can remove the product. It's a constant product removal pulls pathways like the Krebs cycle forward. Specific pathways, you need to create a negative delta G for these reactions to actually occur. You have thermodynamic and kinetic stability. We have glucose is very thermodynamically unstable because there's a lot of energy there in glucose, but it's kinetically stable, which means that it requires physical enzymes to break it down. So glucose is thermodynamically unstable. There's a lot of energy there for heat-wise to generate energy, but it requires kinetic and it requires enzymes because it's kinetically stable to break it down. So that in general is just a brief little overview of that specific topic there. Let me break down this one and show the next part of it. So I'm now going to be going over a ton of specific different science concepts. That way people can watch this, but also re-watch these. I'm going to share the specific screen here that I'm trying to show and look. There we go. Mhm. All right, guys, this is overall a big quick sheet of a ton of specific different topics here. It's for a lot of people that do the medical college admission test, it's just a big overview of specific science. Obviously, biology is a study of life, but how we behave in the environment is also psychology and sociology. Psychologically, how the mind impacts the body, and sociology, how we are actually going to be impacted by this specific environment. These all play a crucial role to our health, but overall, when health goes wrong, the pathology. So there's the nervous system, which is consisting of the brain, the spinal cord, and all of your nerves that come out of that, which is your peripheral nervous system. I remember learning about this. The motor neurons, it requires effort. For efferent, it's going to be coming outside of the nervous system to make things move, work. It takes effort to carry out of the nervous system. There are efferent signals that from the central nervous system can go to muscle. It can go to glands. It can facilitate movement or action, and it plays a critical role in responding to stimuli, which can execute voluntary and even involuntary actions. Voluntary is like what I'm doing with my hand. An involuntary reaction might be when you get hit in the tendon and you have an involuntary kind of like reaction that occurs. That's an involuntary reaction where your motor neurons are being impacted. That's the efferent system that carries out. Interneurons are a big role of your neurons, which are the functional unit of your nervous tissue. Interneurons, they're in between. They make up the majority of the neurons. They're interneurons. They're like the connectors within the central nervous system, process information, sensory inputs, motor outputs, this reflex arc you've probably heard about. Interneurons are essential for complex behaviors, inhibiting specific things, coordinated responses. They're very important. Sensory neurons are on half into the nervous system. These are sensory carrying information throughout the body, throughout the central nervous system. They help to perceive your environment, and they're very important for feedback systems. So sensory is afferent with an A. Efferent is motor. And they come from different parts of your nervous system, like a front region or the front portion, which is closer to like your belly button. It's going to be more of the ventral horn of your spinal cord, a ventral portion. For efferent information, dorsal, like a fin on your back, is going to be more for sensory afferent information. You have your parasympathetic. Para is something that in medical terminology in Latin means beside. So it's just beside the sympathetic nervous system. The parasympathetic nervous system is found primarily in like your lower back and your neck area. In the head region, it's responsible for the autonomic nervous system, which just is ongoing all the time. Like when I'm undergoing peristalsis and I'm relaxing and I'm consuming food and I'm resting on my back, just lying down, the parasympathetic is more active. Digestion, salivation, lowering of heart rate, because it's beside the sympathetic nervous system. The sympathetic is more in the middle part of your back. It's that fight or flight. It's in the middle part of your back because it's in close reaction to the heart. It's starting to increase the heart rate. It will dilate areas of the pupil, but it's going to be redirecting blood flow as your sympathetic nervous system to the muscles for exercise, running around, sprinting. is a very sympathetic movement. Lying down, resting is very parasympathetic. It's more of the calming nature. So parasympathetical, contracting of the meiosis of the iris, contracting of the eye portion of it. Sorry about that. And many different other functions there. Sorry, that was in Spanish. Yes, so the sympathetic nervous system is obviously going to be needing to emergency, survival mode, etc. Yeah, there's organizations of the specific portion of the brain. The brain has different layers to it. It has different structurally looking different. Like the cerebral cortex looks different than the cerebellum. Then you have like the stem of the brain, and there's different portions of the stem of the brain: hindbrain, midbrain, forebrain, etc. Medulla oblongata. All of those different areas can help with autonomic function, like breathing. Heart rate can be impacted by specific brain structures. The hindbrain is responsible for coordination and balance. The medulla oblongata regulates function like heart rate and breathing, like what I talked about before. It's for consciousness, arousal, and consciousness. The midbrain, the middle portion of it, is a relay center for motor signals and sensory signals. And it has the superior colliculi, which I believe are like little indents in that area, which can impact vision and hearing, respectively. Forebrain, for more forward-thinking functions, sensory relay center. The hypothalamus responsible for homeostasis, overall balancing of things, regulating the endocrine system, which is like negative feedback loops. Like an example of day-to-day life is like your air conditioning. It turns on when it's a specific temperature. Yeah, that's kind of, in general, negative feedback homeostasis, which is going to be obviously hypothalamus, which hypo is below thalamus. Coordinates movement. Limbic system is for emotion and memory, and the cerebral cortex is for higher thinking. That's all the forebrain structures. The thalamus is the big relay station for sensory information, which has various inputs for sensory modalities and can direct areas of the cerebral cortex, which is the thalamus. It's the relay station for relaying information all throughout the brain. Hypothalamus is under the thalamus. It plays a critical role in maintaining homeostasis, body temperature, hunger, how hungry you are you thirsty, tired, do you need to sleep, general day-to-day cycles of rhythms that comes from the eye, light, all the way through to create these rhythms via the hypothalamus, because it relays information and it integrates it with the hormone system, the endocrine system, and also the hypophyseal portal system, which is where you have two parts of your pituitary glands: the anterior pituitary gland for the endocrine function, and the posterior pituitary, the neurohypophysis, is for the nervous system part of it. Our body folds and via human development, and specific areas have different functions based off of where it is manifested in the central nervous system. The basal ganglia is a movement regulator. These are a group of nuclei, is a ganglia, a collection of nuclei, a collection of cells that facilitate voluntary movement and help with the regulation of posture and muscle tone. These play a significant role in learning motor skills, etc. That's the basal ganglia. They also need healthy amounts of oxygen to survive for postural activities. The limbic system for our overall memory and emotion systems. We have something called the amygdala, which is involved in fear and aggression. The hippocampus, which looks like a seahorse, I believe, is critical for memory. And then the fornix, which connects various limbic structures. The fornix, I believe, is like a point structure. Some of these names are dictated by what it looks like when you dissect a human cadaver, which I've done before. It's really, really cool. I did it in chiropractic school for about 9 to 12 months. An amazing experience. I'm blessed to have been able to do that. The cerebral cortex, the cortex is like the outermost layer, The cerebrum is the outer layer of the brain. Cerebral cortex is responsible for higher level functions. It helps to perceive things. It helps with general reasoning. It's decision making. If you have a brain injury or a traumatic encephalopathy of the brain, various concussions, head trauma, your perception isn't that good. Your reasoning and decision making aren't that good. Guess what also impacts this? Toxicity via poisoning from alcohol or other things can affect your perception, your reasoning, and your decision making. Can also affect are you thinking of yourself and others, which is your frontal lobe. Parietal lobe, can you actually sense and process what people are saying, which is your parietal lobe. Your occipital is in the back, helps with the visual processing. The temporal, which is by your ear, it actually helps to do with hearing. Frontal lobe, parietal lobe, temporal lobe, occipital lobe, etc. Cerebral cortex, then the cerebellum is there, which we just did talk about that. So there's neurotransmitters that are really important. They transmit these nervous signals. Acetylcholine, I said that's low. Acetylcholine is voluntary muscle control, attention, alertness, and functions of the parasympathetic nervous system. That's acetylcholine. A lot of supplements, you could take choline. Choline is found in egg yolks. Acetylate, add an acetyl group to choline, you have acetylcholine. Voluntary muscle control. The neurotransmitters that release at this kind of neurotransmitter onto the muscle is acetylcholine. Attention, alertness. I like taking choline supplements to help with my alertness and my focus. But also can help to relax you too. In the parasympathetic nervous system, you have norepinephrine and epinephrine with wakefulness and alertness. Caffeine and exercise is going to stimulate your epinephrine and norepinephrine. Dopamine has a positive reward pathway. It makes effort overall just make you want to do it. Serotonin is mood and sleep cycles. Uterine contraction is by serotonin mainly, I believe. Appetite and emotional regulation is done via serotonin. GABA is an inhibitory neurotransmitter for calming. Stabilizing neuronal activity, which you can take GABA as a supplement. You can take most of these things as a supplement too. You get endorphins, natural painkillers. You get this whenever you're with your family, whenever you're exercising, you get endorphins. Like hands-on massage work, I believe, gets endorphins when it works normally. Okay, so we talked about sensation versus perception. You guys can see how the eyeball goes to it connects to the brain via the optic nerve, a string-like system that's in the back of the eyeball that goes to the brain. It connects it. You have from the ears, you have the tongue, you have those equilibrium ear circular canals that help with your balance. You have to get these stimulus into electrical signals, which is the nature of the nervous system, so that way the nervous system can interpret these so the brain can actually make sense of the environment. And it's very complex on how this whole process occurs too. So that was the nervous system, how it intertwines with human behavior, social dynamics, group thinking, how we're going to be interacting with the entire environment. We can learn via habituation. Over time, things can become very habitual. Like your phone alarm when you wake up in the morning can be very habitual. It's done with repetitive stimulus. This is how you can train your dog. This is how you can train your loved ones. Dishabituation is trying to do the opposite of that. Observational learning, like whenever you're watching a video, you're obviously using observational learning. Hopefully this is done in a positive way. You can observe others by watching a video. You can observe others like when you're a baby, observing your parents, observing how people walk when you're a baby, learning how to walk. A lot of this is done observationally to start, and then you're obviously going to do via physical learning. You can be conditioned, like in a specific environment, to behave a certain way, whether it's positive, whether it's negative. That's obviously up to interpretation. But classical conditioning is a learning process that pairs a neutral stimulus with an unconditioned stimulus to get an elicited response, like that Pavlov's dog environment, etc., to associate a bell with food. The neutral stimulus is the bell. Unconditioned is going to be something that you want to get them to do. So it's a stimulus that then can condition them to think bell with food. For a lot of people, this can be you do it with your dog, you do it with your little kids. If they do something positive, then you might give them a specific reward, which is obviously a different set of systems too. Operant conditioning is a type of learning where behavior is controlled by consequences. You can positively reinforce something like that was good, give them something positive that they like I love steak. So if I do something good, I might reward myself with steak. Negative reinforcement, if I do something bad, I might get specifically physically punished, or I might get let's say taken away money, etc. Because trying to shape a behavior. So sleep. Sleep's really important. You have specific stages of sleep. We talked about the nervous system and the functions of the brain. They help with sleep. You need a good cold environment. You need circadian rhythms to be healthy for healthy sleep cycles. Stages of the sleep cycle, you have waves of oscillations of electrical activity, EEG waves, are awake during, so your awake state, you have indicating alertness, which are beta and alpha waves. Theta waves, more in sleep. Stages 1 and 2, theta, delta for deep, 3 and 4. Restorative processes, mixed frequencies during rapid eye movement sleep cycles, we have dreams occur. These are going to be oscillating and repeating throughout the night. Those are the general stages of sleep. You could use your Oura Ring, you could use any kind of fitness tracker. A lot of them track sleep nowadays. Okay, so we all probably have heard about specific pharmaceuticals that can cause depressants. There are things that can be like alcohol, benzodiazepines, SSRIs, selective in reuptake inhibitors for serotonin. They can show serotonin's more available, GABA. These actually inhibit brain activity. Stimulants, coffee, nicotine, amphetamines, Adderall, dopamine and norepinephrine signaling things in the brain. I don't know why I use that photo. Opiates, morphine, powerful pain relievers for euphoria. These can be highly addictive, and hallucinogens, magic mushrooms, LSD, which is psilocybin. These are going to affect serotonin, which is for your mood. Cognition, consciousness, and language development theories. Your cognition is essential for problem decision and making. We, of course, know that. Consciousness, very debatable, even where that comes from. Feelings, perceptions, and states of consciousness. Language development theories, etc. So understanding what parts of motivation, this can be trained, this can be a learned behavior. You can have internal rewards, you can have intrinsic behaviors and intrinsic motivations and extrinsic motivations, extrinsic from outside the body, intrinsic from inside the body. And of course, when your brain is being very healthy, these work good. When your brain is not healthy, they typically don't work good. And you can be very emotionally unstable. Because emotion is a complex psychological state, this is important in medicine and just general healthcare when you're caring for other people, for whomever you are. Emotional well-being is very, very critical because you needs to have a high level of decision making. In your response to stress, it's going to be critical because you can have stress hormones like adrenaline and cortisol all the time are going to be impacting how your emotional well-being is. And also how we identify our own self within our own environment. It's how we perceive ourselves, our current state, our past experiences, and our future aspirations. There are psychological development outlines. Can you trust people? Can you feel autonomous? Do you have shame about yourself or your family around you? This is all going to impact your ability to just generally work in the environment itself. So there are many stages to development, but there are theories that we have pre-conventional, conventional, and post-conventional, illustrating how moral reasoning evolves with our age and our general experience. You've probably heard of like openness, conscientiousness, extroversion, how agreeable you are, and how neurotic you are. Those big 5 theories. I wonder if you've done some of those personality testing. I know a lot of them are very gray as to how accurate they are. I've done various personality tests before. I've done them with careers, with specific jobs. They had me do specific personality tests in plenty of these different scientific science courses. They've had you done them. But I believe some people have also debunked them as well on how accurate they are and how accurate they are. You're probably wondering how we overall form our identity. There's obviously going to be just various different theories and stages, which we just talked about, to be honest. So these are social processes that refer to whether individuals and social norms and behaviors. Key processes includes how we are socialized, which is a lifelong process of how we socialize to our specific environment and the norms of society, like what's good to do for people around you, what's good or bad to do for strangers, what's for the law, what's against the law, what even the law is. All of that are social processes and how we can form attitudes and how we are influenced by our surrounding environment. How cognitively we actually believe these things are right or wrong, and all of these are up to debate. You probably see these when you have people what's the word? They bring signs and they go up to governments and they go and they start to protest. Yes. So you have status roles and groups of these social interactions. Who has the status? What are these specific roles? Should you act in these roles? And then how are these groups formed, etc.? How do these individuals interpret and understand causes of their own behavior? It can involve these big, huge types of how we are attributed and how we are being affected by these specific behaviors. So, how you attribute to things around you, how individuals interpret, and how this relates to their thinking and behavior. This it focuses on the process of inferring the causes of our own and others' behaviors, generally divided into dispositional, internal causes, and situational, external. What disposition are you? Internal, and then external. Stereotypes are cognitive shortcuts that make us have sense of social information. They're often oversimplified, they can be, and they are generalized beliefs about a particular group of people that can lead to a confirmation bias, like confirming what you already believe. This is important for research when you look to try to prove the things that you want to prove. And if things disapprove what you like, you typically don't try to confirm it because of your pre-existing beliefs, where you grow up, what schools you went to, if you went to school or not, etc., what your parents told you, what's right, what's wrong, all that stuff. This impacts the social and structural demographics of all of the world, etc. The how educated, what education you did, the economy, politics, and how this influences your behavior throughout yourself in society. Your age, your gender, your race, your income, all of this what sociologists are going to be trying to access. Nowadays, obviously, this is very important with epidemiology, the study of a lot of things, the study of the public environment. These are general definition terms of the incidence of something refers to the number of a new case of a specific disorder that can occur in a specific population during a specific time period. The incidence of a crime rate can be 1 in 100,000. That would be a specific definition, understanding the risk of contracting something. How prevalent it indicates the total number of cases of a specific thing in a population at a specific point in time. Is how prevalent is it? Morbidity refers to the state of being or diseased of the incidence of a disease within a population, which can also include the complexity of health status and the impact on quality of life. Mortality denotes the number of deaths caused by a disease in a specific population during a specific period of time. Big 180. This PowerPoint is a little bit just in random order. The building blocks of proteins, classifications of amino acids, and levels of protein structure. Already gone over this quite a bit. This can help dictate what I find for some people that if they're not typically well fed, it's going to affect their psychological and social well-beings. Building blocks of proteins are amino acids. Amino acids can be nonpolar, hydrophobic, or polar, hydrophilic, positively charged, or negatively charged. That dictates the function of the proteins. There's primary structures all the way up to quaternary structures of proteins. Simple to more complex. Binding of these proteins play a critical role in transporting molecules across a cell to maintain a controlled environment of homeostasis. Receptors are really important. We talked about neurotransmitters, we've talked a little bit about hormones, but you have to receive these things, and your receptors will detect what signal is being done, which is going to impact what is the structure of these hormones or neurotransmitters that dictates what the receptor can signal or detect. And this will play a crucial role in initiating a specific cellular response, and these can be categorized into different specific structures and functions, etc. Enzymes, they're proteins, but they catalyze. They help to speed up a chemical reaction to help it occur quicker. They lower the needed activation energy of a chemical reaction to occur. These are crucial in various biological processes that help to increase the rate of reactions without being consumed. Enzymes are not consumed in the process. And people study this. There's specific kinetics, the studies of some of these enzymes, how an enzyme's catalyzed and how fast it goes. The reaction velocity depends on the material concentration, the substrate concentration, and the maximum reaction velocity, which is the velocity Vmax of this reaction to occur with an enzyme. You have the maximum velocity divided by how much times how much substrate material there is, divided by how affinity, how strong or how sticky that is to this specific complex of this enzyme that's trying to help this reaction to occur. This is regulated by various factors. Enzymes are going to be impacted by the temperature of what the local environment is, the concentration of hydrogen ions, and the presence of things are going to be inhibiting or activating things. A lot of foods, supplements you could buy, pharmaceuticals are going to be inhibiting things or activating things. You've probably had caffeine before. It's going to make sure that adenosine is still around. Adenosine is a sleepiness agent, I believe. Caffeine makes you feel more awake because it's going to be doing some inhibiting. So inhibiting or activating, it can bind onto specific things, and it's going to affect the receptors. There are different kinds of inhibitors: competitive inhibitors, non-competitive inhibitors, uncompetitive inhibitors. There's mixed ones. Certain inhibitors, like certain pharmaceuticals, can bind to things, like an SSRI, a selective serotonin reuptake inhibitor, can inhibit serotonin to make sure inhibit something that degrades serotonin, which can allow for serotonin to be more prevalent, which can hopefully affect your mood. It can change the enzyme structure and function of these specific inhibitors. Enzymes are, of course, regulated. I just went over that one. So carbohydrates are necessary. They have a specific structure. They also have a specific function. They are organic molecules. Carbohydrates have carbon, hydrogen, and oxygen, CH. Those are they're classified into monosaccharides, one type, disaccharides, two types, polysaccharides, many types. Monosaccharides are the simplest form. Disaccharides are just two monosaccharides. Like sucrose has fructose and glucose, polysaccharides are longer chains of monosaccharides that can be branched or unbranched. These give us various functions in our body, like glycogen. Starch is going to be a sugar, cellulose, fructose, etc. I'm going to be right on back. All right, thank you for waiting. So move this camera right here. Perfect. All right, guys. So we talked a little bit about DNA so far and about all the specific things that it does. DNA is crucial. DNA biotechnology is obviously getting very popular. This is done with so much stuff, checking your hereditary, checking with what specific DNA is found at a crime scene, etc., all that stuff. So DNA is deoxyribonucleic acid. It does not have an oxygen. You've probably heard of like polymerase chain reactions, PCRs, CRISPR's technology. It's used in medicine, agriculture, genetic research. It's getting pretty cool. RNA is ribonucleic acid. Is a single strand of nucleic acid, ribonucleic acids, ribose sugars, phosphate groups. It can have adenine, guanine, cytosine, or a uracil. Unlike DNA, is deoxyribose. Ribo has oxygen. DNA has thiamine, but in RNA, it's replaced with the uracil. You guys are eating DNA and RNA all the time whenever you're eating food. You're getting this genetic code from the food you eat. The genetic code consists of these codons that are sequences of 3 nucleotides, like an adenine or a uracil or guanine. And there are many, many specific codons that can code for specific like amino acids. So our body does use codes, just like the technology that we use. Biological structures, there's a phospholipid structure towards our cell. I'll go over generally cells, has many different organelles. You've probably heard of human development, how we start as one cell and start to divide. Our cell membrane has lipids, which are fat, and phosphates. Phospholipid bilayer, which provides us structural integrity and forms as a selective barrier. Your skin is a barrier. Each cell has their own specific barrier. The presence of cholesterol is found in our membranes and helps to maintain this fluidity and stability of our cell membranes. We also need to transport some of these things that we eat from our food or transport from one area of the body to the next. We have to do this via a passive or an active mechanism. Passive is where you can diffuse, or you can use a facilitator to diffuse, and it does not require energy. And it occurs like down the hill, down the concentration gradient, whereas active transport across from one area to the next, it does require energy to move these substances against, like up the hill, against a concentration gradient. So how can material go from a cell, get out of the body, and go to another portion of the cell, and vice versa? It's done with endocytosis to go in the cell. Exocytosis is done outside of the cell. Cyto refers to cell. Endo is inside. Exo is outside. These are key processes for transporting these molecules, especially large ones. Endocytosis involves the engulfing of the substance that goes into the cell. Like a hormone goes from, let's say, the thyroid gland, it has to go to your for males, the testicles, women, the ovaries. We go via to get in the cell will be a process of endocytosis. Exocytosis is expelling the materials from the cell, like which is useful for neurotransmitters and hormones. Because you have to get out of that gland that makes it for a hormone, or from the brain for a neurotransmitter. It has to be exocytosed outside of that specific cell. So those processes are obviously highly regulated and needs to occur. So how do we get this energy that we need to consume? And how do we get ATP? We can get this via glycolysis. You get glucose, it goes into pyruvate, and we get ATP. This is like a metabolic factory with many different steps. And some steps are reversible and some steps are irreversible. The ones that are irreversible are highly regulated, where the managers are there making sure things are really good at that specific step. This is impacting the bioenergetics, the life energy, metabolic regulation, and how we get ATP. So this is how life and forms work. You obviously have so many different kinds of biology, so many different kinds of work. We have eukaryotic organisms like you and I, prokaryotics like bacteria. There are viruses, which are non-living entities, which we're well aware of about how viruses impact our body. But we have hundreds and hundreds of trillions of viruses on our body and inside of our body. So we are working with them, but obviously they can wreak havoc on our bodies. Same with bacteria, but not all are bad, right? So these metabolic pathways, we need to have enzymes to help with the chemical reactions. You need to have the material, which is the substrate, which is like the food you eat. Enzymes help break down the food you eat, and our body makes enzymes. We need to have a good balance of breakdown of energy, catabolism, the build of energy, which is anabolism. ATP, adenosine triphosphate, is an energy currency. You also have adenosine diphosphate, 2 phosphates, and adenosine monophosphate is 1 phosphate. Having ATP can actually inhibit some metabolic pathways, but having ADP or AMP, like what I talked about, you need more phosphates, so you're going to make those metabolic processes work. These are crucial for biochemical reactions and powering things. So that was sugar. You can break down fat. You can break down amino acids. Lipolysis, the breakdown of fat. Triglycerides are converted into glycerol and free fatty acids, primarily found in our fat tissue. We need to have adipose, but we don't want to have too much adipose tissue. These free fatty acids can undergo energy beta oxidation in the mitochondria to produce acetyl-CoA, which can then enter the citric acid cycle for further energy production. So how do you burn fat? Well, you're typically going to have to consume less energy, consume less energy less frequently, but also burn more energy, have perspiration. Lipolysis can undergo with elevated body temperature. You could break down some of the adipose tissue into free fatty acids that then goes into the mitochondria, which is an organelle of the cell that helps to make energy. You can also metabolize amino acids. They can be metabolized for energy. They can help make sugar. Let's see. These go. They either enter at pyruvate, acetyl-CoA, or citric acid cycle intermediates. You can deaminate, get rid of that amine group. Process removes the amino group, producing ammonia. Breaking down of amino acids can get you ammonia and a carbon skeleton, which can be used to make new sugar as gluconeogenesis, or it can enter the citric acid cycle. So, how you guys make lipids can be excessive energy in general. Fatty acid synthesis involves the conversion of this acetyl-CoA group to fatty acids through a series of condensation reactions, which obviously includes water with condensation, which are catalyzed by fatty acid synthase, an enzyme that makes fat. This is stimulated by insulin, insulin is found after a meal, especially a sugary meal, and it's inhibited by the opposite of insulin, which is glucagon. You've probably heard of glucagon like peptides, GLPs, Ozempic, etc., tirzepatide, retatrutide, whatever it is. They're glucagon like peptides that is going to be inhibited with, so lipid synthesis is inhibited with glucagon, stimulated with insulin. There are amino acids that are essential for us to get into the diet and non-essential because our body makes them. So non-essentials can be synthesized from the body. This impacts their availability for protein synthesis. Essential amino acids must be obtained through the diet, or you can take it as a supplement, etc. All right, guys, so let's talk about the cell. The CEO of the cell is the nucleus. It houses the genetic material, coordinates cell activity for growth, metabolism, and reproduction. That's the nucleus. It's the CEO. The mitochondria helps to create ATP. It has two bilayers to it. It is believed it's coming from bacteria. It plays a crucial role in energy metabolism. So the ribosomes are the sites of protein synthesis. It helps to translate messenger RNA into polypeptide chains that are essential for cell function. So think protein synthesis for ribosomes. There's an endoplasmic reticulum. It's a network of membranes, as seen in the picture, that's involved in the synthesis of lipids and proteins, because some of these endoplasmic reticulums has a bunch of ribosomes attached to them, which ribosomes synthesize proteins, but also the endoplasmic reticulum can synthesize lipids. But it's also involved in detoxification. The Golgi apparatus is like the post office. It modifies, sorts, and packages proteins and lipids. So packages proteins and lipids is the Golgi apparatus. Golgi apparatus is the packager. It plays a crucial role in post-translation modifying of proteins. Lysosomes are like this membrane-bound organelles that contain enzymes that are important for digestion, breaking down waste materials, cellular debris. They're like the trash people. They play a critical role in recycling. Those are lysosomes. Moving on forward with the next steps of how do we get reproduction and development. Very complex. I like watching those videos on YouTube that have animated videos of human development, human reproduction, how we go from one specific cell to fertilize one portion that's a one cell. You see one dot, it divides into a bunch. It divides into a bunch that looks like a raspberry or a blackberry, and then they start to kind of form, exit, then they start to exit a specific area, and then it becomes us. So we produce sexually, and some organisms reproduce asexually, but we reproduce sexually, and we need to get genetically varied offspring to get genetic diversity within our offspring to fertilize. Fertilize and cleavage. Fertilization is the process where the sperm cell merges with the ovum, the egg cell, from a female, resulting in a zygote. Zygotes cleave, it leaves a series of rapid cell divisions, divide, gives us a multicellular embryo, where we have this formation of these layers. After you fertilize, where an embryo undergoes gastrulation, which gives us these specific layers, and these layers form different regions of our body. The ectoderm, outermost, mesoderm, middle, endoderm is the most inner portion. These layers give rise to the organs and the cells that are us as an organism. For example, the ectoderm makes up like the outer portion, like your nervous system, your skin. The mesoderm is like your muscle and some blood cells that move around a lot, like your lymphatic system as well. The endoderm are going to be kind of the inner lining of some of your internal organs, your epithelia. There are stages to this development, from the zygote, the embryo during the early development, and then becoming a fetus. there are specific milestones. I recommend to get good at that, you have to watch those specific videos. Human development is a very complex thing. So homeostasis, the role of homeostasis and hormonal regulation is a biological system's stable internal environment despite any external changes is homeostasis. How to create a stable internal environment, no matter what the external environment is doing. So think temperature, think pH is important, the power of hydrogen ions, how hydrated you are, water levels, mineral levels, and blood sugar levels in the body are all tightly regulated. Your endocrine function, your hormonal glands, like your thyroid gland, they're all undergoing homeostasis a lot. Things that are giving feedback, your feedback loops are really important. So the temperature of the environment you're in a lot, how much you're moving around, how much exercise, what's the temperature of the environment you're exercising in, how much sugar are you drinking, how much sugar are you eating, how much macromolecules and micromolecules are you drinking and eating, etc. This is all going to be impacting the feedback mechanisms within your body, etc. So in general, tips, I like to drink mineral water. I don't recommend you overconsume caffeine. Make sure you don't drink your sugar. Make sure you don't eat too much sugar. Eat what you're 70 to 80% satisfied. I recommend people have one less meal that they have throughout the day. And I recommend people don't consume alcohol and don't smoke. Those are good. And sleep really good in general. And have a healthy romantic life, family life, etc. So we're talking about the heart here. The heart is going to be on the left side of your body, of course. You have 4 chambers. You have atrium on top, ventricle down below. The shape of it is as you see there. So right, left atrium, right, left ventricle. Blood flows through the heart in a specific sequence that's starting from the body into the right atrium, moving to the right ventricle. So RA, RV, up to down to the lungs for oxygenation, and then returns onto the left side to A to V. You probably heard of left AV node, left SA node, sinoatrial node, etc. Those nodes are the electrical activity of the heart. It's being pumped by the left ventricle. And the blood vessels are also helping with the mechanism of pushing the blood to the entire body. The digestive system is responsible for breaking down food. We have a bolus of food, then you have a specific amount of chyme, that is the liquid portion of the cell or of the food that we are consuming, that then has to be digested. Big macromolecules into micromolecules. So, neurophysiology and action potential. So, action potentials are obviously really crucial inside the body. Let me open this real quick. All right, guys. So the neurophysiology of action potentials of how things get from one area to the next is very complex. But in general, you need to have sodium, you need to have potassium, you need to have voltage gated channels and specific pumps. You need a rapid depolarization of a neuron's potential. Just think polar energy, positive and negative end. You got to get that energy to go. You need to reach a specific threshold. Sodium-potassium pump can maintain a specific voltage, but then you need a voltage-gated sodium channel, allowing that sodium ions, Na+, to flow into the specific cell to make it to go from negative to a positive depolarization. Sodium-potassium pump first, always there. Voltage-gated sodium channels. It's a gate that is changing with voltage, positive and negative end. You get the picture. A repolarization phase is going back down. This is going to be with potassium because the potassium is going to then bring that thing back to where it was, allowing the potassium ions to exit the cell, which is going to make it more negative. This is crucial for a propagation, like propelling forward of the action potential along the axon, which is going to be carrying this process out of the neuron. There's an all or nothing principle that states once the threshold is reached, an action potential will occur no matter what. Action potentials are really important for especially whenever we want to have day-to-day function, but dysfunction of the body, dysfunction of people with neurological diseases, you could assume their action potentials are not working very well if they're not thinking sharply, moving clearly, moving like autonomically on their own, etc. Their action potentials are not necessarily working because their strength and speed of what they're doing isn't really working well. All right, guys, the skeleton and the muscular system and the connective tissue, like your fascia, you have bones, muscles, cartilage, tendons, and ligaments. Fascia, these provide structure, they provide support and general movement of the body, and they are a crucial role in things like the muscles can help with blood glucose levels, etc. So, kinematics, how the body specifically and functionally moves. Arthrokinematics, joint and musculoskeletal function. A lot of the joints in the body are kind of like curvilinear. They kind of have a curve to it. As you can see here, the head of this picture here, it's like an egg. And then this thing that's bounded to it is like a spoon. The egg and the spoon need to be generally in the center. That way you can kind of rotate throughout the center. You don't want that egg to sit on one part of that kind of curved spoon. Otherwise, it's just not going to function well. In general, that's a very brief, but also just like not wrong way of how to look at joints. The arthrokinematics of things like the jaw joint, it has to go up and down. This jaw bone has to sit well into that head portion of the temporal bone, and it has to move well. Arthrokinematics is going to be dictated by physics. Obviously, the speed of things, whether things are going to be not well into the joint, whether they're dislocated, whether their position is off, but also the speed of the specific arthrokinematics, etc. So these are very important for athletes, but also day-to-day function. And Newton's law of motion. The first law states that inertia states an object in motion stays in motion unless it's acted on by a specific net force, like exercise, etc. The second law of states that the force, F force, equals the mass times the acceleration. So in sports, for certain sports, if someone has a lot more mass and they're faster, that force is going to be greater, and it's going to freaking hurt. Makes sense, right? But if someone's lighter and slower, they're not going to have as much force. The third law states for every action, there's an equal and opposite reaction. So work is defined as the product of force and displacement, how much one has moved in the direction of the force. Think of like moving a heavy tire or moving a heavy piece of furniture. How much was the displacement? How much was it moved? And the force. And then obviously the angle. Is it a flat environment? Is it an incline or a decline on the treadmill, etc.? And obviously how gravity is playing a role. The kinetic energy is the energy possessed by an object due to its motion, expressed as kinetic energy is half of the mv squared, mass and velocity. So mass is important, the speed of things, of course, kinetic energy, all that good stuff. Potential energy is like stored energy. It's potential due to its position, its structure, its configuration. It's commonly described as like gravitational positional potential energy. How gravity is impacting the body and the height of the person. You've probably heard as we get older, potentially we shrink over time. It could be due to the potential energy of gravity. Hopefully we don't, right? The work energy theorem states that work done on an object is equal to the change in its kinetic energy. Work and energy directly. That's the work energy theorem. So thermodynamics, you've probably heard of the specific laws. The first law states that energy cannot be created or destroyed. That's the first law of thermodynamics. It can't be created or destroyed. It's only transformed from one energy to the next. This implies that the total energy of an isolated system remains constant over time. The second law of thermodynamics states that the total entropy of the system, entropy is the random disorder of things, it can never decrease over time, and energy transformations are not 100% efficient. So that's important. So energy transformations are not 100% efficient. This could be, I believe, with like technology wear and tear, car wear and tear. When you transform energy, it's not 100% efficient. Results, some energy can be dispersed as heat, or in that heat can degrade other things around it. So the second law of thermodynamics is that energy transformation is not 100% perfect. Third law is the processes. No, these are types of thermodynamic processes. They're isothermal. There's constant temperature. It's not change. It's iso. Adiabatic. There's no heat exchange. Isobaric. Baric is pressure, air pressure. Iso, there's just constant. Isochoric is constant volume and having these characteristics. Those are just general terms. H refers to enthalpy as the total heat content of a system. The total heat content of a system is often used by determining the energy changes during chemical reactions. It can be calculated by the delta H, the energy of products, which is like the right side of the equation, or products are like the food that you're trying to make. The reactants are just like the basic ingredients, etc. So it's the total heat content of a system, products minus the reactants. So guys, fluid physics is going to be important because most of our body is water. You consume water all the time. Blood is mainly water. So fluid dynamics is going to be critical in how fluids, liquids, and gases are in motion. Focusing on these forces and these movements, it encompasses various principles, which we have pressure in our body, air pressure, the weather changes, how much pressure is internally in your body, what are you like, how many times are you breathing per minute, etc. That changes the pressure, the velocity, the speed of things, and obviously just the environment, the height, the diameter, etc. of in a fluid flow, like how your blood vessels, how large are they, how large are you, is going to impact the fluid dynamics. The continuity equation states that the flow rate, like the fluid movement, must remain constant from one cross section of a pipe to another. It means it's always dynamic, it's always moving, which is what dynamic means. There are hydrostatic pressure. It's the study of fluids at rest. By Pascal's law, states that pressure applied on an enclosed fluid, like your blood vessel, is transmitted undiminished throughout the fluid. Another principle describes buoyancy and states that an object submerged in a fluid experiences an upward buoyant force, like buoyancy, how buoyancy and kind of springy it is, equal to the weight of the fluid displaced. So fluid weight matters. So obviously, like what is in the fluid, like this mineral water has total dissolved solids. Those solids like calcium and potassium, based off of the periodic table of elements, they all have different weight to them. Which is going to change the obviously fluid displaced, the fluid force, etc., just based off these hydrostatic principles. So with the technology that we're using all the time, you're going to have specific circuits, etc., how light comes out of the environment, etc., all that good stuff. So we have Coulomb's law, which describes the force between two charged objects as inversely opposite proportional to the square of the distance between them. So distance matters. And the product of the magnitude of the charges also matters with electrostatics. Electric potential energy is a uniform electric field that can be calculated by using the U equals QED. We have electric potential energy, Q is the charge, E is the electric field strength, and then the distance. So you have overall strength of the electricity, you have the distance does matter, of course, and the specific charge of that area, etc. So you have Ohm's law, you have V equals IR, you have a voltage, you have I as a current, you have resistance. You've probably seen those triangles here where you can put V, I times R, V divided by I, V divided by R. This analyzes circuit behavior, how outlets can charge stuff, the voltage, what's in there, current, resistance, etc. The resistance to something and all that stuff. So the capacitance is the ability of a system to store charge per unit of voltage. So how batteries hold charge, capacitance, how much these batteries, their capacity to hold the charge. Larger batteries for smaller batteries. There's represented by the formula. Capacitance is the C is the charge divided by the voltage. Q is charge, because it can't be the same thing as C. So Q is charge and V is voltage. The capacitors, which are in so many batteries nowadays, they store electrical energy and they're in various circuits. Electric cars are a big example. Most of our body moves in waves and oscillations. The nature of waves can be classified as electromagnetic based off of the propagation. It's a physical transfer of matter. Sound waves are a longitudinal wave that propagates through a medium like a solid, liquid, or gas by compressing in the particles of the medium. It requires a medium to travel through the sound waves. The speed of sound, it travels faster in denser materials, such as water and solids, as the speed of sound. So we have obviously seen sound waves have a wavelength, frequency, and an amplitude. You've seen the wavelengths. You've probably seen the crest is the number of waves. Number of cycles per second is the crest. Or sorry, well, measured in hertz, cycles per second. And the amplitude is like the power of the wave, how high it goes up, related to loudness for the sound waves. So how many times per second, and then how large for the amplitude. Now, with light, with how our eyes conceive light, how light is coming from my environment, there's a wave-particle duality. Light can behave a wave and a particle. It's the wave-particle duality. This is crucial for understanding the interference and diffraction, as well as the photoelectric effect. How photons can generally create energy and move from a high energy state to a low energy state, etc. And also, like, you know, when you've had glass, the light reflects off the glass. Sometimes you get that prism, all that stuff. You get light refraction. You get something called Snell's law. It describes the refractive index and the angle of incidence and refraction. How light passes from one medium to another, changing the speed and the direction with Snell's law. The total internal reflection occurs when light attempts to move from a denser medium to a less denser medium, and then is at an angle greater than the critical angle. This is utilized in optical fibers for data transmission. It's the internal reflection from a medium to another medium. You're going to have a specific reflection of light. You've probably heard of converging lenses, diverging lenses. Converging is like a convex to a point. It brings light to a focal point, like lasers utilize this. And then diverging lenses with specific eye lenses you might wear is more diverging, it's more opening. A lot of healthcare modalities or machines use converging or diverging lenses, converging to focus, diverging to be like more open. And then I believe it's the thin lens equation, relates to the focal length of a lens and its curvature and its refractive index. So you have the length, what kind of lens is it, well, relative to its curvature, and then how much refractive light there is. The behavior of light, very important, can be described using these geometric optics for rays and wave optics for phenomena, light, diffraction, and interference. Geometric optics applies when light travels in straight lines, while waves optics create wave properties of light. So getting down to more of the level of the atom involves the behavior and interaction of atoms, which is the smallest functional unit of life itself is an atom, because it can't be broken down into more. An atom cannot be broken down anymore. Understanding these principles is good for chemical reactions and chemical bondings. Now, nuclear phenomena focuses on the behavior of the atomic nuclei, like radioactive decay, like Chernobyl. You've probably heard about that. How radioactive decay from a radioactive plant can just happen over time. Nuclear fission, and the nuclear fusion. These processes involve significant energy changes, like from power plants, and they are pivotal in nuclear energy and nuclear medicine. So we have force and velocity for vectors and scalars, mass and speed, measures of central tendency, probabilities. For independent events, the probability multiplies. So whenever you get probabilities, the Cartesian coordinate system is used to graph equations where the x-axis represents the independent variable. So how I like to memorize specific things with x-axis and y-axis is the acronym or mnemonic DRY for DRY. Dependent variable is on the y-axis, which is the up and down vertical part of it, which gives us the results, DRY. And then the mix is MIX. It's manipulatable. It's the independent variable. It's on the x-axis, which is the horizontal one. So the y-x-axis represents the independent variable or mix, MIX, and then the DRY is the dependent result in y-axis. It's the dependent variable. Understanding and being able to do math inside your head is really good for your brain. For some people doing testing, like the medical college admissions test, you actually cannot use a calculator, which is too bad nowadays, because honestly, like, you're going to be able to use a calculator. Technology can do it for you, but yeah. So research design is obviously really, really critical. There's definitely a hierarchy, like a pyramid of research. Meta-analysis, randomized controlled trials are at the top. It's harder to do those, but those are really good. Anecdotal evidence from like what you and I believe from seeing, it's important, but it's at the very bottom. Double-blind randomized controlled trials. A meta-analysis is a study of all these randomized controlled trials, etc., on a topic, yada, yada, yada. Research defines, therefore, validity and reliability. How valid something is and how reliable it is. What are the methods used for determining their sample size, their selection criteria, etc.? Because there's probably some things that they didn't necessarily add to the studies because it wasn't selected well. So the causality is the relationship between cause and effect. What was causing this? Is that cause necessarily related to that specific effect? In establishing this, causality versus causation is critical, right? With a ton of things. Requires more than just correlation and necessitates demonstrating that a change in one variable produces a change in another. Was that thing actually causing that, etc.? This can be achieved through these experimental designs, retrospective from the past, prospective going into the future, longitudinal studies studying people over time, and the use of control groups like taking a pill that's nothing in it versus taking an actual pharmaceutical pill. It's a control group, study groups, all that stuff. Strengthening of your claim, you're obviously going to need to get studies for that. Data analysis and interpretation. So the mean is the average of these data points, which can be heavily impacted by outliers. Median is just that central value. Mean is the average of the data points. The median is not affected by the outliers because it's just the central value. But the mode, it represents the most common data points. It's also unaffected by outliers as the mode. The range measures the difference between the largest and smallest values, and standard deviation indicates how spread out the values are from the mean, which can also be influenced by outliers. Those are important. Mean, median, mode. Probability is the likelihood of an event occurring. Mutually exclusive events cannot occur together, while independent events do not influence others' probabilities. The probability value, or the p-value, indicates the probability of obtaining the observed results, assuming that the null hypothesis is true. A CI, or a confidence interval, provides a range of values believed to contain the true value, reflecting the reliability of the estimate. That's why we show graphs, all these figures that you see. They're essential for visualizing data and interpreting results. Nowadays, with more of these language learning models, hopefully that instead of these boring two-dimensional figures and graphs, we can start to get more animated three-dimensional graphs that I think are more fun, more interesting, and easier to read, easier to see. It's important to analyze these axes for all of those specific things. Hope you guys like that specific video. That goes over quite a bit of things, just in general topics, not necessarily in super in-depth, but just in general topics. So I'm going to do some more in particular here. I'm going to look for some of my other things that I had. Yes, there we go. Thank you for waiting, everybody. Let me get this here. All right, so these are able to be watched at a different point for anybody to observe and see these. I'm going to add this one here. We're talking about, of course, amino acids are crucial for everyone's bodily health, and this is something that I'm passionate about. I do a lot of exercise training, and of course, amino acids are a big topic of it. But amino acids are used in biochemistry a lot, everybody. So amino acids are crucial. There's so many different amino acids, but amino acids, like we talked about before, there's a negative end for the carboxyl group and an amino group, which has a positive group. But this is obviously dictated by the power of hydrogen in the nearby local environment, so the pH. But it's a dipolar ion. It has two poles to it. They're net neutral. They neutralize each other. Then there's this R group, which means it's just resulted in whatever amino acid it is. That's where they differ, is in that R group, that resulting group. There's also a hydrogen right here in the middle, and there you go. You need nitrogen for the amino group. Let's say you get NH3, and then you have a carbon, you have oxygens. There's 20 standard amino acids, and you need to understand these if you want to understand specific science. Oh, sorry about that. Okay, so you guys can see that we just talked about the amino acids there. So there are 20 of these amino acids. In this little picture here, which I like having this, I printed this off. I can see it a lot. These different colors are going to be having a different code just based off of that. And this can be a good thing to see in general. We're going to go over these as to why they're different. So the aliphatic ones, you have ones that don't work with water. They're non-polar. A mnemonic I like to think of is we have glycine, which is one of the most simple ones. Great. Ambitions vanish. Limitations isolating. Future for F, phenylalanine, tired medical professionals. You have alanine, glycine, isoleucine, leucine. Leucine and isoleucine are important for muscle synthesis. They're nonpolar. Glycine is a very simple one right there. Proline has this kind of ring to it, and valine has a specific V at the end. So those are some of the nonpolar ones. These green ones are going to have some specific rings to them. Phenylalanine and tryptophan have rings to them. Tryptophan has 2 rings. Tyrosine has an OH off the ring, and phenylalanine also has a ring. Those are important for neurotransmitter health, like tryptophan for serotonin, tyrosine, phenylalanine for dopamine. Then you have ones like arginine, these blue ones, arginine, histidine, and lysine. Those are positively charged amino acids, or they are basic amino acids. Serine and threonine are pink ones. Serine and threonine are able to be phosphorylated, and they have hydroxyl groups. They're hydroxylic. Cysteine and methionine are yellow for sulfur. Sulfur is typically a yellow compound. They're sulfur-containing amino acids, which is crucial for antioxidants and other things. Then you have asparagine and glutamine. They have amino groups. So some of these essential amino acids, they're essential, and it means that they must be made, they must be taken in by the diet. They're not made by itself. They cannot be synthesized. You have PVT, TIM, HALL. So you have these are the amino acids that are essential that need to be taken into the diet. So we have phenylalanine, an aromatic amino acid. It has a ring to it. Valine has a branched chain amino acids. Threonine, tryptophan, isoleucine, methionine, histidine, arginine, leucine, and lysine. You guys could read those depictions there if you need to understand. Each amino acid kind of has their own unique property. And typically, if you're studying for a test, they typically are going to test you on the unique properties of the amino acids. Amino acids are D form or L form, and it just refers to the structure. D is right for L is I think levo for left, D is right, and it refers to this carboxyl group, I believe. They're grouped by their R groups, which are the resulting groups, are their side chains. This side chain from the amino acid, their R group, is going to be influencing the protein's behaviors. The nonpolar amino acids are typically going to be buried inside the protein core. They're typically not going to be on the outside because they don't have polarity. They don't like to interact with water. So these are glycine, alanine, valine, leucine, isoleucine, methionine, proline. They don't like to interact with water. The ones that have aromaticity or have those big rings to it, phenylalanine, tyrosine, tryptophan, they're primarily hydrophobic, but tyrosine has a hydroxyl group, which can make it its exception for being polar. There are polar charged and polar uncharged. Polar means it has some polarity to it. It can interact with water. Serine and threonine are polar, uncharged. Cysteine, asparagine, and glutamine, polar, uncharged. These negatively charged amino acids, they are acidic. They are aspartate and glutamate. They contain carboxyl groups, but they can release protons, which gives them that negative net charge at that physiological pH because they donate protons. Basic, they like to suck up these protons. They can accept protons, histidine, lysine, and arginine. They have a higher pKa value, which means the protonated versus deprotonated forms, etc. So hydrophobic, it does not like to interact with water. The nonpolar side chains contribute to the overall stability of these protein structures because it minimizes specific reactions in an aqueous or water-based environment because they don't like to interact with water. It's because it minimizes the interactions. Glycine is the smallest amino acid. It's actually found in collagen. It's where it actually occurs in every third portion of the protein collagen. Glycine only has a hydrogen in its resulting group. There's a methyl group in alanine. A methyl is a CH3. Methylation is found all over the body too. Branch-chain amino acids, they just have branches off their chains. You have these as a muscle-building supplement. Leucine, isoleucine, and valine. Isoleucine is a little bit longer than valine, and valine is kind of a little bit shorter. It has like a V-like structure of their branch chain. Energy metabolism. Muscle metabolism are the branch-chain amino acids. Methionine is known for its sulfur, but also it's used for a coding region that helps to start protein. So methionine is found in initiating protein synthesis. And sulfur as well. A thiosulfur linkage. Proline is known for a kink in the backbone of a protein, and it's the only amino acid that specifically does it, because it forms a cyclic structure and can introduce a kink in the protein due to its specific structure. So proline needs to be in some certain proteins, but it can't be in certain proteins because of its kink structure. Phenylalanine has a benzene ring to it, which is a ring with double bonds inside that ring area. It's dictating for the structure and function of it. Phenylalanine is used with dopamine, I believe, as well. Same with tyrosine. It has an OH group in its ring and makes it polar for enzymatic reactions and signal transduction pathways. So tyrosine for dopamine, tryptophan for serotonin, for being a precursor for serotonin, which is also helping with melatonin for mood and sleep, etc. for tryptophan. And tryptophan has two rings, a bicyclic ring, bi for two. The polar uncharged amino acids, serine for hydroxyl groups, which can facilitate phosphorylation. Threonine is also essential phosphorylation, and cysteine for sulfur bonds, sulfur groups for two cycle sulfur bridges for cysteine. Asparagine and glutamine can help transfer ammonia because they have nitrogen metabolism and which is good for protein structures. The negatively charged amino acids, aspartate, donates protons. Glutamate also donates protons. Glutamate is an excitatory neurotransmitter for synaptic transmission. The basic amino acids, you have histidine, which contains an imidazole group, allowing it to be a proton donator and acceptor because it has a pKa, which refers to how much protonated versus deprotonated state. It's really close to physiological pH, so it can be it's a very unique one, which is why it's going to be tested. It's important for enzyme sites. Arginine is also a supplement that's used for nitric oxide for blood flow and vascular function. It has a specific guanidinium group, which makes it a highly positive at physiological pH. Lysine is used in, I believe even immune supplements have lysine for enzymes as well. So pH and pKa, understanding specific pKa values of specific proteins at specific pH levels. It's the protonation state of amino acids that changes their charge and their reactivity. So I like the acronym Don't Eat Hard Cheese, Your Knees Rattle. Arginine has a higher pKa value, around 12, I believe. And then aspartic acid and glutamic acid have around, I think, 4, I believe. So it's a pretty low number. So for predicting ionization rates, pH and pKa is predictable for ionization state of amino acids. Ionizing, like separating, is the fundamental and biochemical application for enzymes, for enzyme catalyst and protein interactions. How amino acids are going to be behaving in different environments is, of course, their pKa values. You might be tested on how to memorize these. So carboxyl group has a negative charge, so it's a lower pKa. Amino group has a positive charge, so it has a higher pKa. The negatively charged amino acids has a pKa of 4, histidine pKa of 6. The basic amino acids is about 10 to 12, which dictates its specific function. The isoelectric point is where there's, let me go back. There's no net charge. To determine this, you have to average the two pKa values that encompass from a positive to a negative end, finding the balanced state of a protonation and deprotonation state. So for some amino acids, you typically have to find the two pKa values that are given. If there's more than 2, then you have to just find the specific average of what specific amino acid and protein you're trying to find. So on a test, there's specific methionine and cysteine have sulfurs. Those are the only ones that have sulfurs. There's specific ones that have rings, phenylalanine, tyrosine, and tryptophan. Histidine is a 5-membered ring with 2 nitrogen atoms, which is going to be different than the other ones. Proline has a ring that's attached to the backbone, and that's the only one. Amino acids with specific charges as aspartate and glutamate are negatively charged at physiological pH 7.4, and then lysine and arginine are going to be positively charged at physiological pH. Each amino acid is unique. We talked about methionine and cysteine having sulfur. The aromatic ones, which are the rings, and then proline, and then addressing their pH impact. So the non-essential or conditionally essential means that you're going to find them in specific body regions. Glycine is in collagen. Proline is introducing kinks in alpha helices. It's good for collagen structure as well for proline. Proline and glycine are found in collagen. Tyrosine for dopamine and thyroid hormones. Cysteine is for protein structure and stability, disulfide bonds. Alanine can be an energy creator, gluconeogenesis, think of alanine. Glutamate for synaptic transmission, cellular metabolism. Glutamine is going to be as a supplement that's for nitrogen transport, healing of the gut, recovery, asparagine. Synthesis for glycoproteins. Aspartate for neurotransmitter, detoxification, energy production. Serine for phospholipids metabolism. Glycine and proline are found in collagen. Tyrosine for thyroid and dopamine. I'm doing this again. Disulfide and glutathione, which is an antioxidant for cysteine. Glucose and alanine for gluconeogenesis. Glutamate is an excitatory neurotransmitter. Glutamine is for a nitrogen shuttle, intestinal fuel, metaphorically. Glycoproteins for asparagine. Aspartate for crucial for ATP is aspartate. Serine is for phosphatidylserine and sphingolipids, which are crucial for a lot of things. So mastering amino acids is crucial, and I hope you guys like that specific video. I hope you guys enjoy some of these specific live streams. That way you guys can watch them at any specific point. Hope you guys like these videos, and I'll see you guys in the next one.